Vice President of Geologists, Jackson Hole. Again, I'd also like to welcome you here. I'd like to ask everybody to pull these out of your pocket, if you would, and put them on mute. Turn them off, or whatever you do, to make sure they don't go off during the program here. It's my pleasure to introduce our speaker tonight, Dr. Gregory Dick. Greg has a BA from University of Virginia, a PhD from Scripps, Institute of Oceanography, and a postdoc at UC Berkeley. And he's been at the University of Michigan since 2008, where he is associate professor and associate chair for curriculum. Some of his fields of study include, amongst others, geomicrobiology, molecular evolution, astrobiology, and life in extreme environments. His research interests are very diverse. They focus on things like recognition that microorganisms have dominated the history of the Earth, playing an intimate role in shaping its chemical and physical properties, which is basically the story for tonight, how we got oxygen, why we are here breathing instead of not breathing. And those interests focus on the interplay between the biosphere and the geosphere, again, which Greg will be addressing at least in part this evening. How microbes drive geochemistry, and how geochemistry in turn shapes the microbial diversity, metabolism, and evolution. Teaches a lot of different classes. He supervises undergraduates, master's candidates, and PhD level candidates as well. Long list of publications and research grants and other types of recognition. But I'd like you to join me in welcoming Greg and talking about the oxygenation of Earth's atmosphere, rotation and day dependent, length dependent. It's a question mark at the end. So, Greg. Back to the beginning here, sorry. Well, thank you, John, and thank you all for coming. Thanks also to John and John for the invitation. I'm always delighted to speak here. You might know that we have this Camp Davis facility just south of here where I teach for a month every year. So it's really fantastic to get to come to this part of the country and teach geology and environmental science. We have a really nice symbiotic relationship with geologist Jackson Hole. John comes down and talks to my students and tells them all about his rich knowledge of the area and Wyoming's energy resources. And John and others have also been participating in some outreach that we do. So we have an Earth Camp. This is a camp for high school kids from underrepresented groups that comes out from Detroit and learns all about the geology of this region and career opportunities in geology. So John and others here have been fantastic in supporting that. So thank you, John. And I'm delighted to participate in this feedback, this crosstalk between the organizations. So I'm going to tell you a relatively new story today. This is something I've just started working on. In fact, it's not yet published. So you're going to be some of the first people to hear about this, and I look forward to your comments and feedback. The punchline I'll tell you right now is that we think that changes in the rate at which Earth has been rotating, in other words, its day length through time, might have had really profound and unexpected effects on how much oxygen was produced in Earth's history. Okay, so that's the punchline. To get us in the mood for this subject, I want to do a little activity with you all. So I'm going to ask you to take a couple deep breaths. And don't worry, we're not going to do yoga or anything like that. But I want you to think about that air that's going into your lungs and the oxygen that's then getting into your bloodstream and binding to your hemoglobin and being transported to tissues where it's being used to produce energy, right? And then that oxygen is getting consumed and reduced to water, which is then a waste product of that metabolism. And you do this 20,000 times every day, right? You participate in this oxygen cycle. And if it wasn't for that oxygen being replaced, we'd run out of oxygen in a pretty short period of time. So thankfully that oxygen is replaced, of course, by oxygen-producing photosynthesis. Get this right here. Here we go. So oxygen-producing photosynthesis is the main source of oxygen on this planet, of course, and it has been through Earth's history. You probably think of the main photosynthesizers as being the ones you can see, the plants, the trees outside, but in fact only about 50% of the oxygen you're breathing comes from visible plants and trees. The other 50% comes from microscopic organisms. That could be eukaryotic algae or these little bacteria that we refer to as cyanobacteria. And these cyanobacteria hold a special place because they're the ones that first innovated evolutionarily this oxygen-producing photosynthesis. Okay, so thank bacteria with every breath that you take. And so that oxygen then serves as this powerful oxidant that we and all other animals, like this mouse here, use to run energy metabolism, right? So we use that oxygen to combust organic carbon, which also, of course, comes from photosynthesis. And then on the other end comes carbon dioxide and water as waste products, completing that oxygen cycle. So that has not always been the case, that there's been a big source of oxygen on our planet. And in fact, for much of Earth's history, oxygen was at too low concentrations to support plant or animal life. So I'm going to tell you a little bit about that history of oxygen in Earth's atmosphere, what we know about it, and what some of the big questions are. And then in part one, I'm going to tell you about some of the work that my lab is doing to try and understand what controls oxygen production by microbial mats, which we think were the main producers of oxygen in the Precambrian. And then in part two, which will be much shorter, but hopefully interesting, I'm going to address this relatively new idea. Did changes in Earth's rotation rate, in particular, did longer days lead to Earth's oxygenation? Okay, so here is a simple view of how we understand the history of oxygen on our planet. You can think of it broadly as having three main phases. There was this first phase in the Archean, where there was essentially negligible oxygen in the atmosphere. Not enough oxygen, certainly for people or for any animals, or probably even for substantial aerobic life of any sort. Then here we had this big increase in oxygen concentrations, what's referred to as the Great Oxygenation event. That took us to about somewhere between 1 and 10%. And I should have said this is as a percentage of current levels. So 1 to 10% of current levels. And then in the Neoproterozoic, 600 to 800 million years ago, we had the second step up in oxygen, which brought us essentially to modern levels. Okay? It's levels that could support large animals like ourselves. So we're not all used to thinking about this time scale, right? 4 billion years. And so to emphasize that, I put here we are humans at the very most recent. And then there's the demise of dinosaurs there at 65 million years ago, right? So these timescales we're talking about are long and hard for us to comprehend. Also note that the Cambrian explosion occurred right in here at 540 million years ago, and one of the theories regarding this diversification of animal life, and especially larger animals into the Cambrian and the Devonian, was that we required these higher oxygen levels. So this oxygenation set the stage for life as we know it. Origin life back here at about 4 billion years. So for a long time we thought this was a relatively simple story because it went like this. Well, these organisms that are able to do oxygen-producing photosynthesis developed right around the time of this great oxidation event at 2.4 billion years ago. They filled the atmosphere with oxygen, and that was the cause of the great oxidation event. However, more recently, there's strong evidence that there were actually whiffs of oxygen before that. Whiffs, meaning short-lived, maybe not global in scale, but strong enough to leave geological signatures that there was free oxygen in the atmosphere, at least regionally, and at least for millions of years, if not tens of millions of years. Okay? So this raises some interesting questions. If oxygenic photosynthesis was in place 3 billion years ago, then why did it take so long to get to this great oxidation event 2.4 billion years ago. Right? That's 600 million years, the same amount of time as it took to get from the Cambrian explosion to now. That's a long period of time. And secondly, you might have also noticed that this is also quite low oxygen concentrations. And why did it stay so low for so long? That's almost 2 billion years there where oxygen was substantial but not high enough to support animal life. So you could maybe condense that into this question about why did oxygen levels remain so low for so long? And one way that this has been addressed is in terms of the type of photosynthesis that was being done. So not all photosynthesis is the type that you know and maybe love, oxygen-producing photosynthesis, where those organisms use water to get their electrons for photosynthesis, and they rip that water apart, and on the other end they release the O in H2O as O2. Right? That's oxygen-producing photosynthesis. But there's another form of photosynthesis that's thought to have preceded oxygen-producing photosynthesis. That's called anoxygenic photosynthesis, not oxygen-producing. It's done by various bacteria, but also these cyanobacteria. And in this form of photosynthesis, the electron donor is not H2O, but H2S. In some cases, iron or other compounds. And so what happens there is that the waste product is not O2, but elemental sulfur. And so the idea is that early in Earth's history, when there was plenty of H2S and not so much O2, this might have been more important. It might have competed with oxygen-producing photosynthesis, thereby limiting the amount of oxygen produced. This has been one explanation for why it took so long for oxygen-producing photosynthesis to overcome the buffers and to fill the atmosphere with oxygen. So the disappointing thing about this type of photosynthesis and about this period of Earth's history is that we know very little about it. I think if you look at this top graph here, so this is again a timeline now from 4.5 billion years ago to present. And this top bar represents what was the dominant type of primary production on this planet, the dominant type of photosynthesis. And for a long time we did not have plants, we did not have modern algae. Instead, we had these microbial mats, right, that were thought to have covered much of the shallow oceans and perhaps even much of the terrestrial environments, the continents. So think of these as little microbial carpets that were conducting photosynthesis, and we have a really good geological record of these in the fossil record in terms of stromatolites. Right? Stromatolites were really abundant during this Precambrian period. So we know that microbial mats were abundant here. But whether they were oxygenic or anoxygenic is less clear. Almost certainly they were anoxygenic before the Great Oxidation event, but there's the possibility that they were either mixed or versatile or both were present during the Proterozoic, so during this low oxygen phase of Earth's history. So these cyanobacteria, the main messages I'm trying to get across so far, that these cyanobacteria are a planetary force. They transform Earth's surface by producing this oxygen. But we don't know very much about how they work under low oxygen conditions. There are a few environments today that are still low oxygen where they exist and thrive. Can they produce oxygen under low oxygen conditions? Presumably yes, because that had to have happened at some point. And another thing that my lab is interested in, but that I'm not going to address today, is what geological signatures, what fossils, what chemical signatures might such organisms leave behind in the geological record? So how do we study such mats? Well, I mentioned that they're not as nearly as widespread as they used to be because these things here, plants, trees, and also animals, now compete with them and outcompete them in most environments. So to find microbial mat environments, we have to go to relatively extreme environments that exclude those plants and animals. Right? So here's a picture I took yesterday. I was with my Camp Davis class up at Grand Prismatic Spring. And so that's an example of environment where the high temperatures exclude plants and animals and allow these primitive microbial mats to thrive. Right? So we have no plants, no animals. Also in many of these systems, like geothermal systems, we have very low oxygen. And we have an abundant supply, as you know from the smell of these hot springs, of hydrogen sulfide gas. Right? That sulfuric rotten egg smell. So this is one analog that many different groups are working on to understand how microbial mats work. It's unrealistic, though, in some senses, is that we don't think that these warm environments are necessarily representative of most microbial mats that lived in the Precambrian. Okay, so we're always on the lookout for other analogs. And the one that I'm going to tell you about today is shown here. So these are also cyanobacterial mats, similar to those that are found in Yellowstone National Park, but these are cyanobacterial mats at the bottom of Lake Huron in the Midwest, near Michigan, where I live. And so spectacular cyanobacterial mats, they cover several football fields worth of area, so they're really large, larger than anything in Yellowstone. And they are there not because of heat, excluding plants and animals, but because they live in a low oxygen water column that excludes fish and other normal Lake Huron inhabitants, plants as well. So I'll tell you a little bit more about this analog. This is in the Michigan Basin, so the state of Michigan here. Lake Huron on this side, Lake Michigan on this side. Our field site is up here in northern Lake Huron, just off the coast of the town of Alpena in northern Michigan. And as you can see from this map, this is a bedrock geology map of the Michigan Basin. So just like this area of the country, Michigan was covered by shallow seas that deposited these sedimentary layers that are shown here in cross-section. And the ones I'm interested in for the purposes of this talk is this layer down here, which was a Devonian layer of limestone and also at sometimes of evaporites. Okay, so as these shallow seas evaporated, they left behind salt deposits, including gypsum deposits. And today, as rain and snow melt seeps into the water and through some of these porous rocks, it then accumulates in some of these Devonian rocks and aquifers. It interacts with the evaporites, it picks up salts like sulfate and calcium, so it becomes sulfur rich. Also in the subsurface, that water becomes depleted in oxygen, and then that anoxic, sulfur-rich groundwater vents into the bottom of Lake Huron in sinkholes. So where sinkholes have formed due to dissolution of limestone, this groundwater reemerges and flows onto the lake floor, where it dramatically changes the chemistry and biology of the Great Lakes. So here's a photo of our study site, taken from an aerial photo taken from a plane. The sinkhole I'll be talking about is located right here, just offshore of this island called the Middle Island in northern Michigan. And again, this is happening through dissolution of limestone and through delivery of waters that were interacting with Devonian evaporites. Again, just a little bit closer to give you a sense for the size of this. Here's a small boat for scale. So here's the sinkhole right here. Here's what it looks like in cartoon. So there's a source of groundwater here. This groundwater coming up from those sedimentary rocks and aquifers that I mentioned. It is actually salty, so it's dense. It fills up the sinkhole and then spills over the sill into a larger sinkhole area. And because it's salty, it really hugs the bottom of the lake. Okay? There's a film of about 10 feet where this low oxygen, sulfur-rich water hugs the bottom of the lake. So here's what the bottom of the lake looks like there. These, again, fish, plants, other normal animals are excluded because of this low oxygen, sulfur-rich environment. And in their place live these cyanobacterial mats. So they're thriving under sulfur-rich, low oxygen conditions. We think that's a fantastic analog for Precambrian cyanobacterial mats. So we've been studying these as an analog to the Precambrian mats that we think might have formed stromatolites. To give you a little bit more of a sense of these, I'm going to try and play a short video clip. Let's see if this works. I don't have volume control here. Let's try that again. This was a YouTube so the music was for the YouTube audience. Spectacular morphologies of cyanobacterial mats. These are cone-shaped cyanobacterial mats that we think are being formed due to formation of microbial gases under these mats. So methane and sulfide are building up under these cyanobacterial mats, and they're of interest to us because these resemble, in some cases, coniform stromatolites that are widespread in the geological record.
What is their height?
So the height on those, they vary. We've got some little guys here and some bigger ones as well, but they can grow to about a foot tall.
And what's the process that actually makes the cones?
Yeah, so we're still trying to understand that. It is some combination of a passive buildup of buoyant gases underneath, combined with microbial behaviors, including movement. They would seem to want to crawl towards the surface and increase, basically maximize their surface area. So it's a combination of microbial behavior to maximize surface area, as well as physical processes with gas bubbles forming underneath. When you look closer at these cyanobacterial mats, what you see is they're, of course, not just one organism, not just one species, not even just cyanobacteria. We have a whole zoo of microorganisms in here. You'll see that these purple cyanobacteria are actually filaments, meaning they're colonial, with many, many cells forming a single filament. You'll also see in here these white filaments, which we affectionately refer to as the white guys. They're also sulfur oxidizing bacteria, so we also call them the SOBs. And so they live right beneath this layer of cyanobacteria, and they play a really key role in the story that I'm going to tell you today, because they shade the cyanobacteria and mediate the amount of light that gets to them. Here's now a schematic of that mat. So we have the cyanobacteria that are on top. We have these white sulfur oxidizing bacteria that are below. And then on top of this, I'm now overlaying the chemistry that we see in this environment too. So these organisms, in some cases at least, are producing oxygen. So this blue curve concentration will be increasing here from left to right. So this would represent high oxygen concentrations. As we go deeper into these sediments, that oxygen is consumed by respiration, just like we talked about. You and I are consuming oxygen. There are bacteria in here that are also consuming oxygen. The other key chemical in here is hydrogen sulfide. So again, that would be the smelly stuff that's coming out of Yellowstone hot springs, occurring in various chemical forms here. It's shown in red. So here we have high concentrations of hydrogen sulfide coming from the sediments. And that's being produced by these organisms called sulfate-reducing bacteria that make hydrogen sulfide as a waste product. And then the sulfur-eating bacteria consume that hydrogen sulfide, as do the cyanobacteria. Just to remind you, this hydrogen sulfide is key because we think this was present at great abundance on the early Earth. And it competed with water for that electron donor for photosynthesis, right? So H2S was competing with H2O. So here we have cyanobacterial mats in the presence of low oxygen and in the presence of hydrogen sulfide. So again, a good analog for early Earth conditions. The other interesting thing we found about these mats early on was when we first did night dives. So usually we would work there during the day. The mats look like this. They're purple due to this purple photosynthetic pigment. But at night, the mats look like this. Right? The white guys, the SOBs, had climbed above the cyanobacteria. They had migrated above the cyanobacteria, completely transforming their appearance. And then when we went there early in the morning, we saw a similar thing. These guys were still there in the morning. And so the implication was, what does this mean in terms of photosynthesis, in terms of oxygen production? So just to summarize this schematically, here's our normal daytime setup with the white guys underneath the cyanobacteria. And over here, the sulfur oxidizing bacteria have migrated. They've moved to the top of the mat surface. So what does that mean? Well, we measured the chemistry of this, and what we found was that at night, the sulfide, the red line here, you can see, has shifted upwards towards the surface. So essentially that sulfide is coming up at night, and these sulfur-eating bacteria are following it upwards. Okay, and this makes sense because with no photosynthesis at night, there's no consumption of sulfide or production of oxygen, so what we call the chemocline, the chemical change, comes up until photosynthesis takes place, so it goes back down during the day. So we have this diel cycle, a daily cycle of chemistry and the microbiology both following and mediating that chemistry.
Is there any parallels with that sulfur process to the deep ocean, where you have the hot plumes and the tube worms and the whole collection of animal?
Yeah, good question, John. So last time I was here 5 years ago, I spoke about deep sea vents, which was my main interest at that point. And there are parallels. So these are very similar metabolically to those deep sea vent chemosynthetic sulfur-eating bacteria. So that is the foundation of the food web at deep sea vents. And so it turns out it's not just at deep sea vents where these things exist, but they're most spectacular there because they occur in these symbioses with animals. Yeah, good question. Okay, so this gives us a fantastic environment to ask questions about what type of photosynthesis are these analogs of Precambrian mats doing, and how is that affected by various light and chemical conditions. So to study these guys, we do some in situ work with scuba diving, but we also collect samples here, and here's an example of one of those, what we call fingers, one of those foot-tall towers of cyanobacteria that we brought to the surface. Here's an example of the mat, more of a flat mat that's sitting on top of its sediment. And so we bring these into the laboratory where we can manipulate their light levels, we can do simulations, we can torture them with various chemicals and inhibitors to probe their metabolism and understand what they are doing, all while trying to keep them under realistic field conditions. And then to measure their activities, this is the expert at doing that. This is Judith Klatt, who just finished a postdoc in my lab. So she was working in my lab, and she brought this fantastic technology over from a German group who specializes in this. These are microsensors, which allow you to measure chemistry, especially oxygen and hydrogen sulfide, at very, very small scales. The tips of these instruments are 10 millionths of a meter wide, 10 microns wide, about a tenth of the width of a human hair. And so that allows us to measure chemistry, especially the production of oxygen, the consumption of oxygen, and the production and consumption of hydrogen sulfide on microbial scales, right, on their spatial scales, their size scales. So you use these microsensors to study these mats in the laboratory under simulated environmental conditions. And so what I'm going to show you are her results from using those microsensors. And so what she's doing is she's measuring these chemical concentrations and then using some really clever experiments where she very quickly turns on or off the lights, she can quantify the amount of either anoxygenic photosynthesis or oxygenic photosynthesis. These two competing types of photosynthesis that we can then understand what might govern oxygen production rates. So here we are at night, the nighttime situation with these sulfur oxidizing bacteria on the mat surface. And of course there's no photosynthesis because there's no light. And we did a diel cycle here where we turned the lights on, so now we have low light. And what you can see, this gray line was where that hydrogen sulfide curve was before, and the red line is where it is now that we've turned the lights on. So what's happened is that we see light-dependent consumption of that hydrogen sulfide, right? And we can do some other controls to show that indeed that was this anoxygenic photosynthesis, which to remind you is using hydrogen sulfide as the electron donor as opposed to water. So instead of producing oxygen, it's producing elemental sulfur. So under low light conditions in this environment, we're seeing only anoxygenic photosynthesis, no oxygen producing photosynthesis. We continue that for a period of time. In fact, it's sustainable to have just anoxygenic photosynthesis under low light conditions. But once we turn the lights up towards midday levels, so high light conditions, what we see is that the sulfide gets completely depleted because there's so much photosynthesis taking place that's using up that sulfide. And then interestingly, after a period of time, several hours into this condition, we see this microbial migration. We see the sulfur oxidizing bacteria, presumably because the hydrogen sulfide is being depleted, they migrate back downwards under the mat. And then we start to see some signs of oxygen-producing photosynthesis, presumably because these cyanobacteria are starting to run out of hydrogen sulfide for photosynthesis. So we're starting to see this switch over to a different type of photosynthesis. Now, we're going to see in the next slide an important effect of these sulfur oxidizing bacteria. They were sitting on that layer. Remember how white those mats look? They were reflective, and it turns out they were reducing the amount of light that could get to the cyanobacteria. Once they migrate here, the reflective layer is gone, the cyanobacteria are exposed to high light conditions, and boom, oxygen-producing photosynthesis. So we see this example of a microbial behavior, these sulfur-oxidizing bacteria and their migrations really controlling the amount of oxygen that is getting produced. Here's a summary of what I just showed you over the whole 24-hour cycle. So what you're looking at here is the amount of oxygen produced. That's in micromoles of oxygen per meter per second. Okay, and here we have our daily cycle from 6 in the morning until, let's see, that would be 10 o'clock at night, right? And so here are the profiles, the location of the profiles that I showed you. The dark was at the beginning, so no oxygen being produced. Here's the low light phase. It's all anoxygen photosynthesis, no oxygen yet. We turn the lights up to a high light phase, but it's not for another 4 hours that those white guys migrate downwards when we get that explosion of oxygen production. Okay? So our key insight here was that we have these microbial behaviors that are governing how much oxygen is produced. And so here's a summary of what I've told you so far. And in case you missed anything or fell asleep, I'll catch you up here. So first of all, cyanobacteria conduct these two different types of photosynthesis, right? The oxygen-producing type that most people know about, and also this non-oxygen producing type. They are making a decision based on the environment about which one to do. And thank goodness they decided at some point in Earth's history to do the oxygen producing one, because this was critical for the development of life as we know it. Second, I've showed you this microbial migration, right? This flip-flop between these two different conditions, which governs the amount of oxygen that's produced. And we didn't expect this. Right? We did not expect these sulfur oxidizing bacteria to be governing the length of the oxygen production period. So this was a really unexpected outcome based on microbial behaviors that we don't understand. And so the other thing is we realized, if we go back to this, that both the length of light, how long we expose these mats to light, and also the intensity of the light. We need enough light to deplete all that hydrogen sulfide. Both of those things are critical for determining how much oxygen is produced. Okay, so I gave this talk, I guess it was last year, at a very similar audience to this. We have this program at Michigan called Saturday Morning Physics. And even though I'm not a physicist, they allowed me to come in and give a talk. So I gave this talk to a public audience, and one of my colleagues here, Brian Arbick, who's a physicist and an oceanographer, saw me say that conclusion just now, and he said, so what effect did changes in how long Earth's days were through time, what effect did that have on oxygen production? And I said, I have no idea, I've never heard of that, tell me more. And so it turns out that this is well known to physicists and astronomers, that Earth's days have not always been 24 hours long, right? And so I'm just now wrapping my head around these mechanisms, I'll confess, but I'll try and explain it to you best that I can. Earth's rotation rate has been slowing down over time. And the reason it's been slowing down is that the moon, as you know, creates tides. And as Earth rotates, those tides are sloshing across the bottom of the ocean. They're moving across the ocean surface and creating friction. And that tidal friction, over time, is having a slight impact. It's causing a slight slowdown in the rotation at which Earth goes around. Okay? This was theory for a long time until we put men on the moon with the Apollo mission. And a little known fact is that Buzz Aldrin and Neil Armstrong also left, besides their footprints, mirrors on the moon. Mirrors that we could shoot lasers at, like this, and get those lasers reflected back to measure very precisely the distance between the moon and the Earth. Right? So they've been doing that since the 1960s. What they found is that the moon is moving away from the Earth at a rate of about 3.8 centimeters per year. The moon is moving away from the Earth at a rate of about 3.8 centimeters per year, and that turns out to be exactly what you would predict if you were to conserve angular momentum to account for the slowdown of Earth's rotation. So this really nailed it, that this is a real thing, that Earth is slowing down, and that we can measure that with observations of how far the moon is moving away from the Earth, and that lines up exactly with what we predict from the physics of orbital mechanics. Because to conserve angular momentum, any slowdown in Earth's rotation has to be counteracted by a recession of the moon, a moving of the moon away from the Earth. Okay, so wow, I did not know about that until Brian, my colleague, told me about it. And it turns out that physicists and astronomers have been working on trying to predict just how much Earth has slowed down through time. Okay, so here is the latest data on that. What we're looking at here, this turquoise color, is day length through time. Okay, so again, here we are today, 0 years ago, and here we are 4,000 million years ago, 4 billion years ago. And so you can see that here we are at 24-hour days at the moment. But if you go back in time, this day length gets shorter. These data points here, these dark data points, are estimates from the geological record, from things like corals and stromatolites, which have monthly or daily or annual cycles in which you can try and estimate lengths of days and how many days per year. Okay? So the model matches up with the geological data pretty well. It suggests that day lengths have been getting longer. Back here at about the Cambrian explosion, we had day lengths of about 21 hours. We had a stable period for much of the Proterozoic, where it was 21 hours. And then we had rapidly lengthening days through the Archean. And it's thought that when the Earth first started spinning, it was at about 6-hour days. So the good news is, if you're patient and can hang around long enough, you'll have longer days, tens of millions of years now to work on that to-do list. Okay, so
how do you get fairly rapid slowing, absolutely steady, and then continued slowing?
Yeah, John, I was hoping nobody would ask that question.
Sorry.
So that gets into physics and orbital mechanics, which I'm still trying to learn myself, but it has to do, we have two counteracting forces here. We have the lunar tides, which are creating friction and slowing the Earth down. But there are also what are called atmospheric thermal tides, which has to do with the daily expansion and contraction of Earth's atmosphere. So the thermal expansion of water molecules in Earth's atmosphere. And I'm told that those atmospheric tides have a counteracting force. They exert a torque on Earth's rotation that actually speeds it up. And it turns out that at 21-hour days, there was a resonance in several different types of atmospheric tides that made those much stronger. And so what we're seeing here is a resonance of the atmospheric tides. The atmospheric tides were stronger, so the torque that was spinning Earth faster was stronger. And during this period, it counteracted the lunar tides, so that we had a stability in Earth's rotation. Yes?
What's the reason for the different shades of blue and turquoise?
Yeah, I'm sorry I didn't explain all the different things here, but as you can imagine, there are many parameters that go into these models, including distance of the Earth to the moon and what is that lunar torque. So these represent ranges of those parameters, sort of the extreme ranges of what we would consider feasible for this model. So, we could argue about which one of these was it, but all the models point to the same trend, right? With rapidly increasing day lengths, followed by a Proterozoic stabilization, followed by another increase here in the Phanerozoic.
What's the red on that chart?
Yeah, the red is temperature, and again, I apologize for not walking you through that. These are temperature effects that affects the atmospheric tides that I mentioned. And it's part of this paper because they're pointing out the potential importance of these global glaciations, as you are probably aware, just after the Great Oxidation event and then here in the Neoproterozoic. Those global glaciations may have had an impact on stimulating and releasing us from this stable zone here. So this is a really active area of research. I've just told you almost all I know about it. And there's certainly uncertainties in here. But this is our best knowledge now. And I think the fact that the geological evidence lines up with these models provides us with some confidence that this is real. So of course the first thing that we did is we overlaid this latest prediction on day length, which is shown here in orange, and Earth's oxygenation, which is now shown here in blue, and we noted the striking correlation between how day length has changed and how Earth's atmospheric oxygen has changed. And so we wanted to do some experiments on our mats to determine if indeed changing day length changes the amount of oxygen that is produced. So we collected our samples, Ida went to the lab, she used her microsensors, and we did experiments where we simulated different day lengths on our mats in the lab. Right? So on the bottom here is that same diel cycle that I showed you before, a 24-hour day. And we're measuring, again, there's that burst of oxygen production in the afternoon after the sulfur oxidizing bacteria have migrated. And then we stepped down day length to 21 hours, like what we would have expected during the Proterozoic, 16 hours, right around the Great Oxidation event, and in the Archean, and then 12 hours earlier in the Archean. What we see as we squish these days into shorter windows is that oxygen production zone also gets squished, and we are able to make a lot less oxygen, even when we, on a per hour basis. And so what's happening here is that the lag, that time period where the white sulfur oxidizing bacteria are reflecting the light, becomes a larger portion of that daylight window. Okay, so this biological lag serves to reduce oxygen concentrations. When we go from a 24-hour day to a 21-hour day, we lose a great deal of oxygen. If we go the other way, there's no oxygen produced in a 12-hour day. If you go to a 16-hour day, you produce some oxygen. When you go from 16 to 21 hours, you double the amount of oxygen produced. When you go from 21 hours to 24 hours, there's an increase in about 40%. So these are big increases depending on day length. So those are small-scale laboratory experiments, right, that we're doing in a lab with a single microbial mat. And so what we've been working on in the past few months are numerical models to try and simulate exactly what's happening in those mats. And I'm not going to drag you through the details here, but I want to tell you about what we did and what we learned. So what we're doing with these models is to try and simulate the biogeochemistry, the photosynthesis, and the other reactions that are happening in these mats with a model. And in that model, we have equations that govern how much oxygen is being produced, how much hydrogen sulfide is being consumed, and we base those models on experimental measurements from our work and from work in other laboratories around the world. So this is our best simulation of microbial mat behavior. It allows us to simulate behaviors and regulation of these different metabolisms and how much oxygen is being produced. Now the nice thing about these simulations is once you have a model like that, you can lay that model onto a real ancient Earth and start asking questions about, okay, if we had mats that were covering 15% of the continents, and we make some assumptions about how much carbon was being buried and how much oxygen was being released, we can start to predict how much oxygen would be produced under different scenarios, right? Under different continental arrangements, under different day lengths. And so we use this to make predictions about how much this daylength effect would matter, and the answer was that it's responsible for the lengthening in daylength is responsible for increasing atmospheric concentrations by about 25% for every 100 million years. Now, these are really rough calculations, I'll admit, and there's great uncertainty in some of the daylength estimates. But what this tells us is that this was a substantial effect, and that we had never considered daylength before, and now it's something that we should look into further. There were two questions here, yes?
Is the cyanobacteria always in a mat, or is there some free forms in the ocean? Because then they would be constrained by that.
That's correct, yeah. So thanks for asking that question. We're focused here on the mats, which are represented in the geological record by the stromatolites. It's thought that they were really important in terms of oxygen production because of their density and because of their location on the either the land surface or the coastal seafloor, because of nutrient fluxes to those mats. So they were gobbling up a lot of the nutrients, whereas the planktonic organisms, the one floating in the surface that do most of the photosynthesis today, were thought to be not as prevalent. So you're absolutely right that we haven't considered the planktonic groups. Other researchers have looked at what's going on there. We're focused on mats mostly because of the importance of stromatolites in the geological record and their presumed importance in terms of oxygen production. But that would be another step, is what effect does the daylength have on planktonic communities as well? Yes?
In your picture of the mats and the change in the daytime and nighttime, I couldn't tell whether some of the white guys went over, all of them looked like some of them stayed below. Is that
There are some that stay below.
about equal amounts?
Yeah, so there's some diversity in those sulfur oxidizing bacteria. And we think that maybe they visually look the same, but what microbiologists have found lately is that looks are deceiving, and that at a genetic and physiological level, there are differences between those. Right? So some of the ones that stay behind, in some cases, they're not respiring oxygen, they're respiring things like nitrate. And so they don't need to follow the oxygen as it moves up and down. Yes, John?
Yeah, I'm struggling with, thinking about mats and how they move through each other. Can you actually see this movement in your labs, and how does a mat move through a mat?
Yeah, they do move. You can see this. They're actually very charismatic organisms, these little cyanobacteria. They are mat builders, and they purposefully build structures to optimize delivery of nutrients and so on through their mats. We have this great experiment for students where we take a mat and we tease apart the mat to disrupt it. And in the course of about 30 minutes, you'll see these cyanobacteria gliding together to reform the mat to build what we call little cyanobacterial towns and cities and even highways between them. So these are social organisms that are able to move among one another and through mats and so on. Okay, so the take-home message here is that we think this is an idea that nobody has really thought about. We think it may have been important in determining the pattern of Earth's oxygenation. So what I hope you take away from this talk tonight is that microbial mats, we know, mediated critical turning points, whether the daylength idea is correct or not. That this is certainly the case in terms of Earth's oxygenation. What we found in our studies of this one analog is that mat oxygen production is strongly tied to the length of light that's available and also the intensity of that illumination. And so this led us to this idea that changes in daylength, changes in how fast Earth has been spinning, were likely critical in determining when Earth attained an atmosphere with sufficient oxygen to support folks like you and me, animals. And so this amazes me that we had to have all these right ingredients to come together on this planet to make it habitable. So with that, I'll stop and I'll acknowledge, I mentioned Judith Klatt is the postdoc who did much of this work. There are also some graduate students involved that are listed here. Collaborators, Brian Arbick, who had this idea at another seminar, and then collaborators in the modeling. These guys are from Germany as well. We do this work out of the country's only freshwater national marine sanctuary. This is a sanctuary that's devoted to archaeology of shipwrecks, but these divers, professional divers, are fantastic in helping us sample and conduct experiments on the floor of Lake Huron. So we really rely on them, and then this research is supported by the National Science Foundation. So thank you, and I'd be happy to answer more questions. Yes?
So I was going to ask about your work in Lake Huron. How deep do you have to go? And is it so deep that the sunshine isn't all that big a factor, or what?
Yeah, so
I mean, you're doing a lot of work in the lab, so
yeah.
wow.
So these sinkholes are interesting in that they occur at all different depths, actually from on land to shallower waters. The results that I showed you today were all from the Middle Island sinkhole, which is at about 75 feet depth. And so we have done experiments measuring the chemistry at those depths, and we do see these diacycles. So we're pretty confident that what we've done in the laboratory reflects what's actually happening out there in the real world. But you're right that depth matters too. This is another thing that I think that geologists and geobiologists haven't thought about enough is, how does the geography of the seafloor matter? How does water depth matter in terms of oxygen production? How does seasonal cycles and therefore latitude matter?
How deep does the light go? I mean, so there must be some depth.
Yeah, so that one's at 75 feet. We have other sinkholes that are at 300 feet, and that is out of the photic zone. And so there we find only the chemosynthetic organisms, no cyanobacteria. So it'd be nice to find another one at maybe 150 feet to really nail down what's going on at those depths and light levels.
So all we have to do is slow down the Earth's rotation and we'll take care of all the CO2 in the atmosphere.
That would be
your breath.
geoengineering at an extreme. Yes, sir.
Have you been able to produce H2S synthesis in a long time?
We have, yeah.
Can you repeat the question?
The question was, have we been able to reproduce this H2S-based photosynthesis in the laboratory? And Judith Klatt, the postdoc there that I have described her work, is a real whiz with culturing these bacteria. So she's been able to take single species of bacteria, bring them into the laboratory, and then study that anoxygenic photosynthesis in the lab. Yeah, so there's all sorts of cool stuff that she's done with those laboratory experiments. Yeah. Yes?
So the first oxygen episode was when cyanobacteria started photosynthesis. Am I following that right?
Yeah, except that there's some
sort of.
Yeah, except that there's some evidence, and I'll go back here while I'm talking. There's some evidence that cyanobacteria were also present well before that, right? So we had these UFs. So it wasn't just that cyanobacteria were there, something else had to happen to stimulate this great oxidation event. And I should say that there are other good theories about why that was, right? Geological theories, there are things like there were changes in volcanism, which affected the delivery of reducing gases, which reduced the sinks for oxygen. And I don't discount that those still played a role. I'm just saying that in addition to those, this daylength effect should be considered.
Okay. Well, then I was leaning to, then what about the second episode? And also, what about the precipitation of iron and change in pH
Right.
in the ocean and all of that?
Right. Yeah, so as you know, or as you are likely referring to, we have beautiful precipitation of Van Iron formations through much of this, indicating that we had ferruginous oceans, and that that iron was either reacting with oxygen in the atmosphere, right, was the traditional theory, or I mentioned there's also another type of anoxygenic photosynthesis that uses iron.
Okay. Yeah.
So in that case, you have anoxygenic photosynthesis oxidizing the iron and depositing BIFs in the absence of oxygen. So that's still consistent with this theory. What we've learned in past years about this Proterozoic period is that the oceans were dynamic in time and space. And so there were periods when they were iron-rich, and there were periods when they were hydrogen sulfide-rich. And also it's thought that probably the open oceans were mostly iron-rich, and the coastal oceans were more hydrogen sulfide-rich. So we think that the mats would have been more in the coastal oceans, the shallower depths, where hydrogen sulfide was the main electron donor for photosynthesis. You also asked about what we call the neoproterozoic oxidation events, the NOE. And so I'm suggesting that the map world and these lengthening days might have had something to do with that, right? Because we had this jump, we left that long stasis in the Proterozoic, and that's also when we see this NOE event. In fact, a lot of our modeling is based on that time frame. Again, there are other good theories about what happened here. One of them is that this is also when larger eukaryotic algae evolved. And so there's an idea that maybe they were doing photosynthesis and those larger algae sink better. So they deliver carbon to the seafloor, and to make oxygen you have to bury carbon. Right? And so they're would have increased carbon burial rates that could have stimulated this new proterozoic oxidation event. Yes?
So the iron-based photosynthesis, that would have reduced the iron oxide, right? Liberated oxygen. So
It actually would have oxidized iron 2 to produce iron 3, which would have formed iron oxides or hydroxides.
so that would not have been liberating oxygen.
That's correct. So yeah, this is a big change. We always used to interpret BIFs as a sign of oxygen, but not necessarily. John?
You know, it's always been said that volcanoes were a very significant contributor to oxygen. With this new theory that you're working with, what's the balance in your mind?
Yeah, so we haven't gotten there yet. We would like to quantify what role did volcanoes play and what role did daylength play. These are literally our first stabs at trying to quantify what portion of the oxygenation was this daylength effect responsible for. And so going back to our modeling results, I said that this daylength effect is responsible for about an additional 25% of atmospheric oxygen per 100 million years, right? And so you can see, this is a bad time to look at this on, but this is 1 billion years, so think of tenths of this as being 100 million years. So if we consider this range, the neoproterozoic oxidation event might have played out over 100 million or a few hundred million years, perhaps. And if that's the case, our estimate would be that the daylength effect is responsible for about 50% of the oxygen increase. So there's still room for volcanoes, and certainly I think back here is where it's predicted that we saw big changes in volcanism. Changes between subaerial and submarine volcanism and the amount of producing masses.
Now, the other thing you said is that light intensity is important. So that then begins to get to atmospheric conditions, clouds, types of clouds, and that sort of thing.
Right.
Been talking to any of those
Yeah,
guys?
there's another frontier. Yeah, that's what I love about these new ideas, is there's all these new things to think about. So you're right, and especially if we think about, there's the faint young sun.
Yeah.
And so how did Earth stay warm in a faint young sun? It had this organic rich haze of methane and so on. So there you have two things. You have a fainter sun, so lower light, but also you may have had a very rich atmosphere that also blocked light, so lower light levels. So that's another dimension of this that we haven't considered at all, but I think is a good idea in terms of how light levels impacted oxygen. Yes?
I was thinking too, if the salinity of the water would change the intensity. And as that changes, switching it.
Right. I'll jot that down and maybe we'll do some. That'll be the next idea for the next paper. Every seminar there's another idea. Yes.
A minor point, but you mentioned ocean tides, but there's also southern tides as well. Do you know if the ocean tides are more efficient at slowing the rotation?
I know that only because I'm told that.
Okay.
So the physicists tell me that ocean tides and this tidal friction
The sloshing is more effective than just the...
is not the only, but the main driver of this slowdown in Earth's rotation.
I'm fascinated by the fact that the major changes in the Proterozoic, early Proterozoic, seem to be controlled by immense glaciation. Surely that would mean, presumably, shallower seas across the planet. It would mean reflective light off the ice caps. It would mean all sorts of other sediments coming into the system. So I'm just wondering, it seems to me that if you solve the problem of how that happens, you have a lot greater insight into why that flat zone has appeared for, what, 1.5 billion years.
The problem of the glaciations.
The glaciation.
Yes,
It's so clearly associated.
I agree with you. And another reason, it's thought that rapid changes in atmospheric temperature, like what we would have following a glaciation, had big impacts on the atmospheric tides, which of course are an important component of controlling daylength. So another interesting aspect of this is all related. It's all intertwined. One of the things that makes this fun.
Other questions?
Are there other mats in other places in the world that people are studying?
Good question. Yeah, that's
Lake Erie.
yeah, that's one that I often get from my colleagues that say, is this really representative and how widespread is this? Lake Erie, so I'm studying Lake Erie for a different reason, which is that they're the cyanobacteria troublemakers, right? They form these giant blooms that produce toxins and shut down cities' drinking water supplies. But those are not mats, those are blooms that float on the surface of the lake. There are good mats like this in other sinkholes. So in Florida, right, another limestone sinkhole area, there's some really good cyanobacterial mats that exhibit some of these same characteristics. There's another location in Sulphide Springs in Italy, the Forsassi cave system, where we see very similar characteristics. And so I have a review article that talks about some of those locations around the world. And we do see similar dynamics.
So Greg, that 15% map figure for the cratons, the continents, 15% of what? Which gets to do with the controversy about how big the continents were, how fast they grew.
Yeah.
And why 15% of whatever you chose?
Yeah, so here we encounter, again, pretty big uncertainties in terms of continental area, how those continents were arranged, the amount of coastline that was available. So I presented you with 15% because we think that's a reasonable guess. Honestly, that's all it is, a reasonable
Play
guess.
with different
But
percentages.
we play with different percentages, so we have those sensitivity analyses. We think 15% is good based on what we know about both coastal stromatolite-like structures as well as biological soil crusts. So whereas we used to think that the continents were largely sterile, we now think that these types of cyanobacterial mats covered many soil environments in the Precambrian and also many terrestrial lakes and rivers and so on.
Based on organic analysis of the sediment.
Yeah, and also weathering profiles, paleosols.
Well, I must say, it's not every day that we get to see the absolute cutting edge of some new ideas. And I don't know about you all, but I find this extremely exciting, and that it's kind of probing into a whole new way and an idea of how our atmosphere formed, which is kind of important. So, you know the normal drill, but if you just join me in thanking Greg for an absolutely delightful.
So the normal drill is chairs on this side against that wall, chairs on this side over here against the perpendicular. We'll try and move the curtain back.